Hi Year 11s, welcome to the second week of biology, stage 1. We're going to be looking at just overall general cells this week, so we're going to be looking at comparing prokaryotic and eukaryotic cells. In this PowerPoint you're going to need to be drawing both of those, and I would encourage you to be drawing them by hand. While the exams and everything are going online for biology this year, you still have to have that skill that you can draw cells. So let's begin with the PowerPoint. So this is the need to know for this week. As you can see, some of it is a little bit of repetition from last week when we're talking about cell theories, but the rest of it coming along down here, this is all new information talking about prokaryotic and eukaryotic cells. Now when we say new information, we are also suggesting that this is probably a bit of repeat information from year 8 and 9, but new for the year 11 curriculum. So starting with a bit of revision, looking at cell theory. So cell theory was proposed in the 19th century by 3 German scientists, and there are 3 core principles that we need to know for cell theory are that cells are the fundamental unit of structure of all living things, new cells will only arise from pre-existing cells through cell division, and cells will contain hereditary material, which we call DNA, and that is always passed on to new cells through cell division. Moving on, we're now going to be looking at prokaryotic cells. So prokaryotic cells, they're classified as what's known as a unicellular organism. Now again, in biology, like we said last week, a lot of the things that we talk about, even if you're not familiar with the term, you can work it out. So the word uni meaning one, cellular meaning cell, prokaryotic cells are single-celled organisms. So all prokaryotic cells that we find will be classified as single-celled organisms. The key feature about prokaryotic cells is that they're defined by their lack of organelles. So that's including nucleus. There is no nucleus in a prokaryotic cell. Some examples of prokaryotic cells that you might need to know: archaea and bacteria. Bacteria is the main one that you should probably focus on there. Prokaryotic cells are very small in size, so they're micrometers, 1 to 5 micrometers long. A micrometer is 1,000th of a millimeter. They can also be classified based on their shape, and most bacteria are classified based on their shape. So there's 3 different shapes that we should know, and you'll see them in their name. So for example, if you see a bacterial name and it's got any of these 3 terms in it, you can probably determine what that shape of the bacteria is going to look like. So if they're sphere-shaped cells, they'll be called cocci. If they're rod-shaped cells, they're called bacilli. And spiral-shaped cells will be called spirilla. Now we're going to focus on drawing a rod-shaped bacteria. Now it doesn't matter what one you draw, you just have to be able to draw a prokaryotic cell and label a prokaryotic cell. So over here we've drawn a rod-shaped bacterial cell, so a bacilla-shaped cell, and there's some key features that we need to note on there. So for example, first off is the cell wall. So the cell wall is this thicker green line connected to the fine hairs of the thin lines all around. This thicker green line is the cell wall. Now when you're drawing a cell like this and you're drawing a cell wall, it's important that you show that extra thickness because the cell wall is thicker than the cell membrane. So you can see here I've got the cell membrane in the very light line, and the cell wall is a much thicker line here. So always draw the cell wall as a thicker line. Now in a prokaryotic, you're not going to see cell walls in all cells. They will be in all prokaryotic cells. And the reason for that is that prokaryotic cells are single-celled organisms. They don't have it. Everything they need is in the cell itself. They don't have a skeleton or any external protection. So the cell wall is that little bit of extra structure that the cell has, prevents them from bursting. It's made of polysaccharides and proteins, and it just gives us that structural support for the cell. Next off, so we do have a cell membrane as well, but we won't talk about that right now. We'll talk about that when we get to the other eukaryotic cells, because we'll talk about that in more detail there. But next up we're talking about is the cytoplasm. So the cytoplasm is the fluid, essentially, all the white space here, is fluid inside the cell, and that's going to contain enzymes, proteins, salts, anything that's going to help with metabolism. It's in the cytoplasm. We also have a capsule. Now the capsule is not actually labelled on this diagram, but you can find a good version of the capsule in your textbook, and that's because the one in your textbook is a 3D diagram. It's a lot easier to show a capsule on a 3D diagram. The capsule is basically an adhesive covering around the entire cell, and it helps them to stick to those surfaces. So it's basically the outside of the cell. Pili has a very similar function. So pili is these small lines coming off the cell wall around this prokaryotic cell here. And the pili are small fibrous hairs that are going to help attach to surfaces of other cells. So again, they're helping with attachment. Next up we're looking at the flagella, which is this tail-like or whip-like structure at the end of the prokaryotic cell. Now, not all prokaryotic cells are going to have a flagella. The purpose of the flagella is for movement. So it can move around and propel bacteria through fluids and allow them to move, because this is their entire organism. If there's no resources where they're at, they need to get where the resources are. Now, obviously, not all prokaryotic cells are going to have flagella. They might have other methods of movement. They might stick to other substances, etc. So if they're not needing to move, or if they're not in a fluid substance, they're probably not going to need a flagella, and the prokaryotic cell won't have one. Next up is the nucleoid, which is this region here. Now, we haven't actually got the nucleoid shown on the diagram because they're generally shown as a lighter colour on electron microscopes, but again, it's a lot harder to see a lighter colour on a physical 2D drawing when it's white. But basically what the nucleoid is, in a eukaryotic cell there's a nucleus, and the DNA is stored in the nucleus. Because there's no organelles in a prokaryotic cell, there is no nucleus, and the DNA is just kind of free floating in the cytoplasm. The region where the DNA is stored we call the nucleoid. So it's not a specific region, it's just the area where we can find the DNA in a prokaryotic cell. Lastly, we have the plasmids. So the plasmids, this small circle over here, DNA plasmids. These are special molecules of DNA that can be exchanged between prokaryotic cells. It's one of the ways that bacterial cells have learned to fight off viruses. They'll share the genetic code of how to fight off a virus through that DNA plasmid. But it's really important for genetic engineering too, and we'll talk about this a little bit more in week 6 or 7. But the DNA plasmid, essentially, this is a really useful tool that's allowed us to manipulate bacteria to create insulin for us, which has helped us fight off or improve the fight against diabetes. Moving on, away from prokaryotic cells, we're now going to be looking at eukaryotic cells. Now, eukaryotic cells are much more complex than prokaryotic cells, and they're considerably larger. Part of the reason for this is we believe that eukaryotic cells have actually evolved from prokaryotic cells. Again, we will talk about that a little bit more in the curriculum, just introducing the idea now. The reason that we say eukaryotic cells are more complex is because they're compartmentalized. They contain membrane-bound organelles, so basically little compartments that have their own specific roles, almost like mini organs inside the body, but inside the cell alone. Now, eukaryotic cells can be unicellular or multicellular. So, for example, we are made up of eukaryotic cells, so we would fit under multicellular organisms. Something like yeast, that's used for bread and wine making, that's going to be bread and beer making. Sorry, that's going to be unicellular. It's a single-celled organism. So, eukaryotic cells can be either unicellular or multicellular. Generally, they'll contain the same organelles. So, most eukaryotic cells are going to have the same organelles. There are, of course, some minor differences. So, for example, plant cells are going to have chloroplasts, and they're going to create their own sugars from simple organic molecules, whereas animal cells don't need to create that. We get all our sugars, etc., from digestion and eating other substances. We won't have chloroplasts. In this unit, we're going to look mostly at plants and animals, so let's begin with an animal cell. Again, I would recommend you draw this. Again, I apologise for the drawing. This is a hand-drawn diagram by me, so it's not the best, but it does get the point across. Looking at the organelles that we need to be able to label. So starting with the nucleus, inside the nucleus is the nucleolus. We're focusing on just the nucleus at the moment. Nucleus stores our DNA, and it controls the actions of the cell. We then have our endoplasmic reticulum. There's two different types of endoplasmic reticulum. We have the rough endoplasmic reticulum, and we have the smooth endoplasmic reticulum. Now, the difference between those, essentially, is the rough endoplasmic reticulum is rough because it's got ribosomes embedded into it. And the reason for that is the rough endoplasmic reticulum helps us to synthesize proteins, and it does that through the ribosomes that are embedded in it, because ribosomes help to synthesize proteins to the site of protein synthesis. Smooth endoplasmic reticulum on the other side doesn't have ribosomes, and that's because it's responsible for synthesizing lipids that we use to help make the cell membrane. Next off, we have the Golgi body here in green, and the Golgi body, or Golgi apparatus, either name's fine. The purpose of that is it helps to modify and transport proteins and lipids. It's basically a bunch of membrane sacs, which we kind of see the fact that it's just a whole lot of membranes drawn together. Next off, we have the mitochondria. Mitochondria is the site of aerobic respiration. It helps us produce energy for the cell. Really important. From now on, you're not going to refer to the mitochondria as the powerhouse of the cell, as it's just not a good scientific phrase. The mitochondria is the site of aerobic respiration. Next up, we're going to have our plant cell over here. So plant cells will contain all of the organelles in animal cells, so the ones we just looked at, plus a few extra. So the few extra, so just quickly as well, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi body, mitochondria, nucleus, nucleolus, but it also contains chloroplasts, which is this green one here, which is the site of photosynthesis, helps us produce energy-rich molecules from carbon dioxide and water, because plants don't eat, they have to make their own food. And also the main other difference is the vacuole. And the vacuole helps to store water and nutrients in the plant cell, helps to provide that structure, because again, plants don't have a skeleton or anything like that, so they need the vacuole to store water, help the plant stay rigid and stay upright. You'll also notice there's a cell wall on plant cells, again, just for that extra structural support. It's nothing like a skeleton to protect the plant. Now, the last two slides in this PowerPoint, we're going to look at the similarities and the differences between prokaryotic and eukaryotic cells. So we're going to start by looking at the similarities. As I mentioned before, prokaryotic cells are roughly 3.5 billion years old, eukaryotic cells are 2.7 billion years old, and we believe that prokaryotic cells have given rise to eukaryotic cells through evolution. And because of that evolutionary history, we believe that they have quite a few similarities because of this. So one of the first is that both prokaryotic and eukaryotic cells do contain proteins. Proteins are really important for chemical reactions. They're used as enzymes to help speed up those reactions to make sure they occur at a reasonable rate that supports life. They also have other functions which we'll talk about in a later unit. Both prokaryotic and eukaryotic cells have amino acids, which is really important because the proteins that we talked about up here are made up of amino acids. And if you don't have amino acids, you can't make new proteins. So both cells have to have amino acids to help produce proteins that they need. Similarly, ribosomes. Now, this is where we highlight a little bit of a lie that you've been told across science across the years. Now, we always say prokaryotic cells don't have organelles, but they have ribosomes. And ribosomes are technically classified as an organelle. The difference is they are not a membrane-bound organelle. So prokaryotic cells don't contain membrane-bound organelles. So ribosomes they still have. And the reason that they have ribosomes is they are the site of protein synthesis. So we need to have ribosomes so we can turn the amino acids into protein. So all three of these kind of work together. If you don't have either of those three, you can't create proteins and the cell can't function. So prokaryotic and eukaryotic cells all have those three. Nucleic acids. All cells have some sort of genetic information which is stored in nucleic acids, DNA or RNA. It is stored differently in each cell, so prokaryotic cells contain their nucleic acids inside the nucleoid, whereas eukaryotic cells have it inside the nucleus. All cells will have a cell membrane. The purpose of that is a semi-permeable barrier. It allows us to transport materials in and out of the cell and allows the cell to replenish nutrients and resources that it's using up. All cells have cytoplasm, prokaryotic and eukaryotic, contains enzymes, salts, other important molecules to help the metabolism of that cell proceed. And lastly, both prokaryotic and eukaryotic cells have ATP. ATP is like the currency of energy. Basically, it's a molecule that stores energy that the molecule will release when the cell needs energy to be used inside. Lastly, let's follow up with some differences between prokaryotic and eukaryotic cells. First off, we're going to look at the size. So prokaryotic cells are less than 1 to 5 micrometers, whereas eukaryotic cells are 1 to 4 micrometers long. So you can see here that eukaryotic cells are quite a bit bigger than prokaryotic cells in terms of their diameter. DNA is a big difference. Prokaryotic cells will have one single circular DNA in their nucleoid, whereas eukaryotic cells will have at least 2 DNA chromosome or 2 chromosomes, and they have to be what's considered linear, and that will make more sense as we move through the topic. There are no membrane-bound organelles inside prokaryotic cells, but eukaryotic cells do have organelles. Prokaryotic cells can only be unicellular, whereas eukaryotic cells can be unicellular or multicellular. That's it for this week's work, so that's the theory we're going to cover in week 2. Continuing on this week, we're going to be doing some experiments in class with some microscopes. We're going to be looking at some prokaryotic and eukaryotic cells, determining their size, seeing if you can see any of these, determining differences and similarities between the two. And you'll also be working on chapter 1.2 in your workbook. See you in class.